Hi, it's Mrs. Vance again, and this is the last part of Notes for Unit 10. Today we're talking about how gene expression is regulated. Now, gene expression is basically how the genes, the DNA in the gene, codes for molecules that are going to produce the phenotype that exists as a result of that inheritance. Gene expression needs to be regulated because you don't necessarily need all genes active at all the time. You need to be able to turn genes off and turn them on when you need them. Turn them on when you need them, turn them off when you don't need them. And so we're going to talk first of all about how this works in prokaryotes, and then a little bit about how it works in eukaryotes. As you might understand, as you might accept, that gene regulation in prokaryotes is a little bit simpler than it is in eukaryotes. So it's been studied a lot in bacteria like E. coli, and what they found was, when they first discovered this back in the 1960s, that E. coli often has clusters of genes, and other bacteria as well, that have related functions, and they call this cluster of genes an operon. The operon, for instance, might have genes for all of the different molecules that are needed to make a particular kind of enzyme or a particular kind of product. Each one of these operons has control sequences that are part of the operon, and these include promoters and operators, as well as repressors. So promoter is like basically signaling this is the beginning of this. The promoter is where the RNA polymerase is going to attach. The operator is where the switch is located to turn things on or off. And a repressor protein is one that's going to stop the function of the of the gene. Some operons are what we call repressible. That means that they're on when the substrate is present, and they are repressed or turned off when the substrate is not present. Okay. Others are inducible, meaning the operon is only turned on when the substrate is present. So it doesn't have to be repressed, it just only gets turned on when the substrate is present. So the control sequence is basically the section of DNA that helps control the operon. The promoter is where the RNA polymerase attaches. The operator is the on-off switch, and the repressor is a molecule that binds to the operator and physically blocks or stops the RNA polymerase from attaching. I'm going to also assign an Amoeba Sisters video that does a nice job of showing how this works. So here's the first one we usually talk about, which is, I think it was the first one that was discovered, is the lac operon. Lac operon is the operon that enables the bacteria to break down lactose sugar. Lactose is a disaccharide sugar that is found in milk and other kinds of milk products. And the bacteria need to have monosaccharides to use for cellular respiration. And so being able to break down lactose when you have it present is a good thing for bacteria, especially bacteria like E. coli that live in our gut. So when you drink milk or eat ice cream or something like that, you have a lot of lactose in your system. The bacteria would like to be able to use that, but it has to be broken down. Now, they don't have milk in their environment all the time, so they don't need to make the enzyme to break down lactose all the time. They only need it when lactose is present. So this is a repressible operon. It is repressed unless lactose is present. So when there is no lactose in the environment, the repressor attaches to the operator, and it physically blocks the RNA polymerase from attaching to the promoter. And therefore, it can't transcribe the genes and can't make the proteins that are necessary for this particular process to happen. When lactose is present, lactose actually deactivates the repressor. It attaches to the repressor, deactivates the repressor, makes it fall off, basically, of the operator. And so then the RNA polymerase can bind to the promoter and transcribe the gene, which can be translated into the enzymes that are needed to utilize lactose in the environment. Relatively simple, and there are other genes like that are present. This is just one example of one. It's the most common one that we talk about in terms of prokaryotes, because it's very commonly found in a lot of prokaryotes. In eukaryotes, it's going to be a little bit different because, again, eukaryotes, number one, are generally multicellular, and you've got a lot of different processes going on. You have a lot of differentiation that occurs. And so there's a specific program of expression that responds to various developmental signals in the development of an embryo. The reason we have different kinds of cells is because the gene expression is turned on or off in different kinds of cells related to various factors that exist in the developing embryo. Okay? So what are some things that can affect selective gene expression? Something called DNA packing. This is sort of like what you see in the formation of the Barr body and exon activation. In other words, the DNA can be packed tightly so that it blocks the gene expression and prevents the RNA polymerase from attaching to the DNA, so those particular genes in that particular type of cell cannot be expressed. There are also various kinds of chemical modifications, chemical groups like methyl groups or acetyl groups, that can be added to some of the amino acids or nitrogen bases that can either prevent gene expression or enhance it, turn it on, make it happen more quickly. Both of those things are present in various places at different times in different And I'm going to end these particular notes at this point, and I'm going to turn the rest of the gene expression information over to the Amoeba Sisters. So be sure and watch the Amoeba Sisters video that follows this one.